Hi everybody, I'm Mr. Ayler, and today we're going to learn about meiosis. Summary of meiosis, okay? Meiosis is a process of nuclear division that reduces the number of chromosomes in the new cell to half of what was in the original cell. Meiosis produces gametes, which are sex cells. Cells divide twice during meiosis, and diploid cells undergoing meiosis result in 4 haploid cells, whereas in mitosis, 2 diploid cells would be produced.
Before meiosis can occur, interphase has to happen. In interphase, DNA is replicated, cell processes are carried out, and cells are growing. There are 46 chromosomes in chromatids, and they become 46 chromosomes in 92 chromatids because chromosomes are counted by the centromeres.
The first stage of meiosis, or meiosis 1, consists of prophase 1, metaphase 1, anaphase 1, and telophase 1.
In the first stage of meiosis, known as prophase 1, the DNA coils tightly in the chromosomes. The spindle fibers, they look like this, start to appear. The nucleolus and the nuclear membrane disassembles, and every chromosome lines up in its homologue.
Synapsis is the pairing of homologous chromosomes, which only occurs in meiosis and does not occur in mitosis. A tetrad is each pair of homologous chromosomes. Chromatids within a homologous pair twist around each other. They then break off and attach to adjacent chromosomes.
Crossing over increases genetic diversity. Genetic recombination is the new mixture of genetic combination created. We will now do a demonstration of crossing over.
Okay, so the second step of meiosis is metaphase 1. In metaphase 1, tetrads randomly align along the midline of the cell dividing. Sully, shut up! I'm trying to do a presentation. Alright, whatever. The spindle fibers from one pole attaches to the centromere of the homologous chromosome. The spindle fibers, they look like this kind of, from the opposite pole, okay, attach from the homologous chromosome.
The third step of meiosis is called anaphase 1. In anaphase 1, each homologous chromosome moves to an opposite pole of the dividing cell. The random separation of homologous chromosomes is called independent assortment.
The last stages in meiosis 1 are telophase 1 and cytokinesis 1. Chromosomes reach the opposite ends of a cell, and the original cell produces 2 new cells, each containing 1 chromosome from each homologous pair. Each new cell contains half the number of chromosomes and the original 2 copies as a chromatid.
Meiosis 1 was fun, but now meiosis 2 is boring because it's just a repeat of what happened. Okay, so we started with one cell with 46 chromosomes, and we went through meiosis 1, and now we have 2 cells, each with 46 chromosomes.
Okay, so we're almost through. This is where mitosis 2 starts, okay? In mitosis 2, it begins after the nuclear membrane reforms in the new cells.
The first step of meiosis II is prophase II. In prophase II, the spindle fibers start to form.
The next step is metaphase 2. Chromosomes move to the midline. Remember, metaphase and midline end.
All right, for anaphase 2, the chromosomes that are in the midline, okay, they start to pull away and move towards the opposite cells. So for this one, you can remember anaphase and away. They both start with A.
The next stage is telophase II. In telophase II, nuclear membranes form around the 4 new cells that were created.
The final step of meiosis is cytokinesis 2. The 2 cells that were created from mitosis 1 are now split into 4 cells, each with half the original amount of chromosomes. Now you'll see a demonstration of all the cells splitting in meiosis.
Okay, so what we learned in this video is that meiosis is split up into 2 sections, meiosis I and meiosis II. We began with 1 cell with 46 chromosomes and ended with 4 cells, each with 23 chromosomes. Meiosis creates haploid cells, which are also known as sex cells.
All right, guys, I want to be in a classroom where everyone knows what meiosis is, and I hope that we accomplish that. Sally, I mean, you probably don't understand it, but thank you for watching this pitiful video. You know, I hope you liked it. I did. And thanks to the real Tad Ayler for inspiring this TAD Talk.
Deficiency, so put it in the